Hello and welcome everybody to today's Fireside Chat. This is going to be a new format, and we're here with our guest Gavin Hirst. Hi, Gavin. Welcome to this Fireside Chat.
I'm really looking forward to it, Dennis. Thanks.
Yeah, and welcome to all audience members as well. Thank you for dialing in today. Let me now properly introduce you, Gavin. So for everybody, Gavin doesn't, of course, need much introduction, but we'll go through it anyways. So Gavin is a seasoned biopharma executive and medicinal chemist with over 30 years of experience in drug discovery. And he worked across a range of therapeutic areas, including oncology, immunology, metabolic, and infectious diseases. And he has a track record of delivering high-quality clinical candidates and is, for example, the co-inventor of vaborbactam, which is an FDA-approved beta-lactamase inhibitor that's typically used in combination with meropenem for the treatment of complicated urinary tract infections. Most recently, Gavin was actually the CSO at Atomwise and was leading the efforts in AI and machine learning small molecule-based drug discovery. Prior to that, he was the interim CSO and senior vice president of chemistry at Turning Points, which of course is a precision oncology company that was acquired for $4.1 billion by Bristol Myers Squibb in 2022. He was also, prior to that, the head of immunology chemistry at Janssen, and there he was involved in numerous immunology assets that went to the clinic. For example, lorlatinib, which is a GI-restricted pan-JAK inhibitor that advanced to phase 2 that we'll hear a little bit more about today. Gavin was, prior to that, the VP of Discovery Research at Takeda, and there he directed the portfolio in immunology, oncology, and metabolic diseases, and that led to multiple IND filings for Takeda at that time. Prior to that, Gavin held multiple leadership roles at AbbVie, GSK, and SGX Pharmaceuticals. Gavin is currently executive advisor to the board of directors at Zephrans. He's also a member of the Talis Bio Scientific Advisory Board and an advisory board member of Intelligence Ventures, which is a VC firm that's investing in AI healthcare opportunities. Gavin earned his PhD in synthetic organic chemistry from the University of Southampton in the UK under Phil Parents, and completed postdoctoral research at the University of California at Irvine with Larry Overman. Gavin has authored more than 100 publications, presentations, book chapters, patents, and patent applications, reflecting his deep commitment to advancing the drug discovery and development. And we're really glad to have you here today. What an impressive CV this is, Gavin. Welcome again to today's Fireside Chat.
Thanks, and thanks to you and Drug Hunter for the invitation for this new format. Looking forward to it.
Yeah. So let's dive right in. Gavin, can you maybe talk to us a little bit more about what inspired you to start a career in drug discovery and how you first got involved in the field of drug discovery?
Yeah, so it really started for me when I was doing my PhD. All the first year graduate students were expected to give a presentation on some area of organic chemistry that they were particularly excited about or interested about. And many people took that as an opportunity to talk about the most recent heroic synthesis from the Koryo-Danishevsky group. But a postdoc in another group gave me a medicinal chemistry book. And I took it home and really read it in fascination. And one of the chapters that really caught my attention was a story about the discovery of histamine H2 antagonists for the treatment of gastric ulcers. And as I was reading through it, I came to realize for the first time how synthetic organic chemists can modify structures, and that modification results in the manipulation of biological functions. So we obviously understand that as SAR. And it came full circle, interestingly. After I finished my postdoc, I joined Glaxo or GSK in the UK. And in the lab next to me was John Bradshaw. And some of you may know that John was one of the co-inventors of ranitidine, or Zantac as we know it, an H2 antagonist in the GSK portfolio, and at the time actually was the biggest selling drug on the market. So, for me, that really brought it full circle.
Wow, awesome. Yeah, that's very inspiring. And yeah, maybe you touched on a few molecules already. What's your favorite molecule, Gavin, and why?
Undoubtedly, for me, it's ibrutinib. For those of you that don't know, ibrutinib is a BTK inhibitor. It was approved by the FDA, I think in 2013, maybe 2014, for mantle cell lymphoma and other malignancies. There's a really interesting story behind that. It was initially designed by Celera Genomics as an irreversible covalent active site modifier through a active site cysteine. And they built it really not in mind of this being a drug, but really as a tool to help them pull apart the role of BTK in B cell biology and B cell receptor signaling. But as they started to profile it, they realized it actually had a really favorable therapeutic potential. They saw molecules with this modality having high on-target potency. They also not just had potency, but selectivity. And for me, perhaps the most interesting thing, because of the fact that it was an irreversible molecule, it had a durable pharmacodynamic response. So this advantageous mechanism really put ibrutinib, head and shoulders above efforts that have gone before in the covalent space, in my opinion. And it also served to challenge the dogma that covalent modifying molecules were flawed molecules. And it really, I think, it's paved the way for a broader acceptances in this space and really heralding a new generation of covalent inhibitors. And we heard just a couple of weeks ago from Adrian Gill on utilization of an aziridine warhead, which targeting the KRAS G12D mutation. So I think, it's front and center in our armamentarium right now. And I think ibrutinib kind of broke that mold for many people.
Yeah, no, that's really such a fascinating molecule, and I think for everybody who wants to read up on the history a little bit, there is this book For Blood and Money, I think it's called.
Yeah, fantastic book.
Yeah, I would recommend that to everybody as well. Yeah, Gavin, maybe can you tell us a little bit for the audience, maybe medicinal chemists or people that do want to become medicinal chemists that are just starting their careers and, in this landscape of, where a lot of buzz happens around AI and where it's a very multidisciplinary landscape. What type of advice would you give people that are just starting out their careers?
Yeah, I think I'd give a few pieces of advice, but where I would start is you really have to adopt a growth mindset. As Dennis highlighted, you're now in a very multidisciplinary environment. There are new technologies, platforms, disciplines you get exposed to, so you have to really work hard, I think, initially to start to understand the levers that medicinal chemists can pull, but also start to, understand a little bit of target biology and also how you modulate molecules to drive high quality pharmacokinetics. And it's those multiple disciplines that really come together in a cross-functional way that you really have to really embrace that cross-functional piece of drug discovery to go from being a skilled synthetic organic chemist to a seasoned medicinal chemist. It's not just the chemistry part, it's the broad part of drug discovery and all the other disciplines. AI brings an additional piece to that, of course, not least of which you have to develop additional relationships. So now it's important to develop relationships with AI and machine learning data scientists, chemoinformaticians, and computational chemists beyond just the molecular modeling side of computational chemistry. There's been a lot of chatter, I think, is this the end of medicinal chemists as we know it? Now we're going to do all drug discovery by AI. And no, I don't think it's the end of the medicinal chemist, but I do think it's fair to say, for those that kind of resist utilizing artificial intelligence in their drug discovery efforts, I think they're going to be at a real disadvantage in the digital world. And so you really have to bring machines into the loop and blend AI and machine learning technologies, really with human experience. And I think that drives the best outcomes. And I really encourage people to come at it for drug discovery in general from a hypothesis data-driven drug discovery mindset, and particularly leveraging matched molecular pairs. And so you have a hypothesis about a molecule that will solve a particular problem. You make that molecule, and when you get the data back, how does that inform you? You now have a pair of molecules. How does that inform you on the potential of the next molecule to make? And one final piece of advice, and it's I think a pet peeve of mine, is don't just drive potency. There's so many more challenging problems to address as a medicinal chemist than potency.
I love that one particularly, Gavin. Yeah, it's not about just one property. It's like kind of a Rubik's cube, I like to describe it as, right? You have to
Yes.
optimize like multiple different properties at the same time. So yeah, thank you for that advice. Gavin, I was wondering, what do you most like about Drug Hunter?
Yeah, I've really grown to love the Flash Talks. I find them as a phenomenal education resource, and I watch and oftentimes rewatch them. I think there's an unmatched breadth and depth in there that I continue to learn new facets of drug discovery. And so for me, it's the Flash Talks.
Awesome. Yeah, thank you so much. That's very flattering. All right, Gavin. So I want to dive in a few more like, specific strategies and technologies with you today. So during your long career, what are some of the most impactful technologies you've seen emerge? And, how have those impacted medicinal chemistry and specifically like small molecule drug discovery?
I've seen so many technologies over the 30 years. Maybe I'll just choose three. And I think the first one for me is fragment-based drug discovery. For me, was a really impactful technology, and harkens back to the publication by the Fezzik group, I guess in the mid 90s on SAR by NMR. And so they were able to use small fragments and screen them by nuclear magnetic resonance. And I think this really revolutionized hit and lead discovery, and it really set the stage for an explosion of fragment-based drug discovery. People were able to leverage a discipline of high quality, small but efficient binders, and then actually, build them with precision from a low molecular weight starting point. And I think in a testimony to that, I think there's probably 8 or 9 approved drugs currently from bona fide fragment-based drug discovery efforts. I got my first real deep exposure into it at SGX Pharmaceuticals, which was a leading fragment-based drug discovery company. And I was there in the mid 2000s. And the way that they approached fragment screening was to do fragment screening by crystallography. And you imagine as a practicing medicinal chemist that your starting point, hey, we're going to work on this project, we're going to run a screen, and the first thing you see is a picture of your liganded starting point in the protein of interest. I mean, what could be better for a medicinal chemist? I mean, the discipline continues to evolve, and in addition to the more traditional fragment-based drug discovery approaches, we're now seeing an explosion using covalently modifying fragments, and many companies have been founded on the back of that approach. So that would be my first. AI and ML, I think, is going to be probably a transformational set of technologies in drug discovery. It allows us to analyze vast data sets, uncover patterns that would be next to impossible for humans to even begin to conceptualize. There's tons of potential here to accelerate drug discovery programs, and development as well will be impacted by utilization of AI and ML. I think often underappreciated is not just the potential for acceleration. I've seen many instances where AI has really been able to help improve the quality of the assets, and that's equally important. And then the third one, I think it has to be targeted protein degradation. With the evolution of PROTACs and molecular glues, the druggable proteome, I think, has significantly expanded. We're able to approach transcription factors and scaffolding proteins and targets that were really difficult to approach historically. So I think those would be my top three, Dennis.
Yeah, no, those all sound great, and yeah, they're all, as you highlighted, really evolving technologies. So yeah, I was wondering what approaches to drug discovery are you personally most passionate about, and why?
I love that word passion. I often tell people if you can work in an area that you're passionate about and you're good at, I mean, that will be a very satisfying career. And I've done that in my career. And one of the things that has been, I think, part of my genome has been structure-based drug design. So using three-dimensional structural information to help guide and solve project problems. And it goes back to my time at Glaxo when I saw my first liganded structure of a molecule I'd made bound to the target of interest. I mean, I was totally hooked at that point. And going back to what I said earlier about how exciting is that. And of course, there's been massive advances in the technology around X-ray crystallography to generate these liganded structures. And more recently, the ability to get into higher molecular weight proteins using, excuse me, cryo-EM. And I would say probably 60% of the programs that I've worked on have been structurally enabled. And in fact, I've made career choices to keep that front and center because I find it so passionate. It's hard to describe, the energy you feel when you've got a set of really skilled designers in a room looking at a 3D structure and bouncing ideas off each other, and the ability to design precise molecules and see distances to drive enthalpic interactions. Not only that, there's a potential, particularly if you have off-target liganded structures, to drive selectivity. You can see how the differences between your target of interest and an off-target, you can modulate that using structural information. And then equally important, I think, is the ability to help solve ADME problems. And so if there's a functionally silent part of the molecule, in other words, doesn't contribute to potency in the crystal structure, that may be a handle to help solve some of the ADME problems. Sites of metabolism, for example, you may be able to block those if they're functionally silent in your crystal structure.
Yeah. Oh, yeah.
And I touched on fragment-based drug discovery. It continues to be a passion of mine. I think there's so much upside to fragment-based drug discovery. You start with small libraries, so they're readily accessible, maybe 1,000, 3,000 molecules. And the hit rate, in my experience, it can be quite high compared to common approaches. And so I've run many kinase programs in my time, and typically the hit rate there we see 3 to 5%. So you've got a really good selection of starting points. The other thing not to underestimate is you bind to multiple protein sites. You're not just perhaps binding to the orthosteric site, but the multiple binding sites. And we heard a great story from Pat Walters just last week on linking fragments that were close in space around a binding site, and they were able to link those fragments. And it's certainly an area of fragment-based drug discovery that can be impactful. Not only can you bind to multiple sites close to the active site, you can bind to allosteric sites. And there's a great story from Novartis on their BCR-ABL program, starting from a fragment-based drug discovery, but targeting the myristorylation site using fragment-based approaches. And it really allows the DNA de novo handcrafting of the molecule, allows you to really build them carefully. Someday I'd love to hear a talk from Dan Erlandson from your SAB. Obviously, it's a space he knows very well.
Awesome. Yeah.
Probably the third one that I'm passionate about is property or probability-based drug design. Really, it's a careful focus on physiochemical properties. And I mean, I think it's pretty clear in this day and age as well that utilization of AI for property prediction will really add some muscle to that and probably generate really high quality predictions. we heard Patrick Schinder talk about this a few weeks ago, I guess middle of last year, and one thing he focused on was use of lipophilicity, and that's something that really is always on my mind. We all know we can drive potency by building lipophilicity into our molecules, but there's a huge penalty you pay for that privilege. You're ending up in high ADMET risk space. High lipophilicity correlates with low solubility, and if you have low soluble molecules, it really makes things challenging doing cellular assays at higher concentrations. Going in vivo, it impacts your bioavailability. Even when you're getting to tox studies, your ability to get to multiples over your on-target IC50 to execute on a tox study is problematic if you have low solubility molecules. High lipophilicity correlates with high clearance. So that's clearly not something you would want in your molecules. And it also correlates both with high promiscuity and high tissue accumulation. So if you've got high promiscuity in multiple tissues, that's clearly something you need to avoid. And then the last one is AI in this space. I think there's 4 advantages. Scale. I mean, undoubtedly, it's unbeatable. You can screen hundreds of billions of molecules from the enamine real library, or probably even trillions of molecules from the Explore library. It's just a scale that's unapproachable by other techniques at the moment, virtually screening. You can screen virtually any targets, even targets with no ligands and no structures. And I think AlphaFold 2 has really helped with that part of the puzzle, but really you can approach multiple targets. And you can do this using out-of-domain extrapolation and using foundational global models that really helps you get there. Speed, I mean, at its simplest, I guess I would say all things being equal, it's just compute power. And obviously with the emergence of DeepSeek, it's going to be interesting how that really impacts the ability there. The hit rate as well, and I think is something to talk about for virtual screenings using AI. And I draw people's attention to a publication in Nature Scientific Reports last year by the Atomwise company, who had shown in the paper that they screened, I guess, about 320 diverse targets spreading the broad proteome, and the hit rate there was about, I think, probably 75%. And even with those that were dark targets that had no ligands, no structures, their hit rate was pushing 70%. So significant advantages of using AI in that setting.
Yeah. No, you touched on AI a little bit already. Can you maybe talk a little bit more about how AI changed your approach to hit ID and lead optimization compared to like the traditional methods? And how do you see the emergence of AI impacting small molecule drug discovery maybe in the next 2 to 3 years?
Yeah, I think I kind of touched on the first part there. But going forward, I think that there's certainly high excitement in this space, and a couple of things that personally I would look for. I think one is a continual enhancement of generative AI synthesizability into the algorithms and the models. One of the current weaknesses, although there are organizations trying to address this, is that generative AI often will recommend structures that are impractical or not even synthesizable. So really doubling down on that. There was a great publication in JMed Chem focusing on that this generative AI has to be able to incorporate synthesizability access and evaluation into that algorithm. I firmly believe that all organizations will end up bringing some elements of AI into the design, make, test, and analyze cycle. I think it's inevitable, going back to my earlier comments about moving into this digital space. The other thing that I would love to be able to do, and people are working on prediction of tox endpoints, but I would love to have enhanced capabilities, not just from using data from a stable of molecules, but doing it from the minute you draw a molecule on a piece of paper, that you're able to say, aha, at least from a binary standpoint, a binary classification, this molecule has a high potential of being cytotoxic. This molecule has a high potential of being genotoxic. Make this, not that. And so you're looking at, to be able to narrow down your choices and make the highest probability molecules. So that for me would be, a really, really big impact, be able to do that.
Yeah, that'd be really amazing to have that. Yeah, no, we talked a lot about different technologies now. So I want to get to your experience with different projects a little bit more. So maybe, you have worked on so many different projects during your career, even had, many drugs in the clinic and had approved drugs, which not many of us can say that they have an approved drug. So I would love to hear your perspective on what do you believe are the top characteristics to consider when you bring a new project in your portfolio?
Yeah, I think oftentimes there's a rush to, well, let's start building a target product profile. But where I start is, you have to align with corporate strategy and a TA focus. That's really where you have to start. You have to know your organization. So it's when you're thinking about things, it's going to align with internal capabilities, maybe some proprietary platforms you have or proprietary technology. I think it's particularly true in biotech. So really start with knowing your organization. When it comes to a deeper dive into potential new projects, I think there are many areas, but there's some key ones that I think about. Do you really understand the unmet medical need and what is the standard of care out there and how are you going to differentiate from the current standard of care? That has to be a big part of how you think about bringing in a new projects. How are you going to think about patient selection? What is your patient selection hypothesis? Is there an ability to identify biomarkers and target engagement markers to help with the translation into the clinic? Is another piece that I would typically think about. One that's often underserved is really getting your arms around target-based risks. What do we know about on-target toxicology? And in this day and age, with the advent of CRISPR, we can start to really ask those questions. And of course, traditionally we've used knockout models, and we can certainly talk about the value of those. But really getting your arm around target-based risk. Probability of technical success is a big one there. How does it align with your internal capabilities and the challenges up against the target you've taken? I mean, how are you going to have starting points for a small molecule problem? Are you going to start from a known compound, and so that you're now likely in a best-in-class discussion? And yet again, going back to my earlier comment, you really need to then really understand what a differentiation strategy needs to be against what's ahead of you. And if you're starting from an unknown, you get to start a project identifying a new ligand. You have the potential of being in a first-in-class situation, but obviously you're then bringing on a larger set of unknowns because you're blazing the trail in an area. When you're first in class, you're having to really learn things that haven't yet been learned. So your starting points become impactful there. And then the final piece, and these two go together, is the competitive landscape and the market potential. Now you're going to be third in class or fourth line therapy. That those are things that you have to think about deeply from a competitive standpoint.
Yeah. And you touched on this already a little bit, but how do you think about like balancing high risk targets where you're probably going to be first in class if you get an approved drugs versus like more established strategies where maybe there's already a drug on the market, so you're going to be maybe best in class if you go, and then balancing short-term project goals with long-term strategic visions for the drug discovery pipeline in your experience as an executive?
Yeah, this can be very different in a smaller biotech than in a larger pharma. Obviously, the larger pharma has a toolkit that a smaller biotech doesn't have, and also has almost always a broader portfolio. And so how you approach it there can be quite different. I think in general, what I've tried to do is harmonize it around two areas, and that is ensuring that you continue to drive innovation. And this kind of speaks to more to the first in class, and balance that with a sustainable portfolio of lower risk programs to really balance that. So innovation and sustainability, I think, are really important. And to allow you to do that, some of the practical things that I've used in the past is, you have to have some clear stage gate decision making, set milestones, know what success looks like. And it may look quite different for more challenging targets than, well, better known targets, better characterized targets that are best in class. But you really have to have an understanding of what success looks like. And you also have to come at it from a position of, what I sometimes refer to as dynamic portfolio management. You may see that a high risk program is actually moving far more quickly than you'd anticipated. So it really allows you to reallocate resources or funding if you're really doing a dynamic project management or portfolio management. The other thing really allows you to do, if you're doing it in a dynamic sense, is really to respond to changes, respond to emerging data that may deprioritize or apply a higher priority to one of the targets. Even allows you to respond to emerging landscape changes. And I think one of the things that I think sits with me is, the high excitement with the first KRAS G12C inhibitors coming to the market, such high potential. But what was shown is there was a more rapid emergence of resistant mutations than was anticipated. And the revenue stream from that was not as anticipated. And when you see those things going on, and if you're working in the KRAS space or have programs in that space, it allows you to think carefully about continuation or refocusing of those. So that, dynamic portfolio management. And the last thing that I've tried to do, talking about innovation and having some of those high risk targets, is oftentimes it can be better to do those in a collaborative sense with other organizations that have capabilities you may not have, doing it with smaller biotechs, doing it with public-private ventures, doing it with academic groups, really to help manage some of that risk and bring in techniques and technologies you don't have in-house.
That definitely makes a lot of sense, yeah. And maybe, Gavin, can you walk us through a particularly impactful project that you've led and talk a little bit about the key success factors that, made this project so impactful?
Yeah, I'm going to talk about the discovery of lorputinib, which was a GI-restricted pan-JAK inhibitor that I worked on at J&J. And this was for the treatment of inflammatory bowel disease, so ulcerative colitis and Crohn's disease. And the reason the team had focused in on a GI-restricted approach was twofold. One, of course, we know where the target organ is. It's in the GI tract, so doesn't it make sense to be right where the target organ is? And so that was one piece of that. And the second part was that it was well characterized that maybe all current JAK inhibitors carry a black box warning. And so if we had a restricted molecule that had minimal to zero systemic concentrations, we felt it would give us high confidence we'd be able to avoid those adverse events. And as we sat and thought about it, we knew we needed two things. We knew we needed low intrinsic permeability. You didn't want to passage through the GI tract and into the systemic circulation, but you had to do that low permeability while still having high cell and tissue activity. And that seemed like a bit of a conundrum to us. So how to bring that together? I go back to my earlier point about hypothesis-based drug discovery. The hypothesis we adopted was one, well, we intuitively felt there must be a physiochemical space out there that captures that almost unicorn space of low intrinsic permeability, but with high cell activity and tissue activity. So what we did, we set about trying to define what that space might look like, doing it experimentally. The team started with a known permeable pan-JAK inhibitor from the literature. And part of the reason we chose that as a starting point was The crystal structure and modeling information showed that it had a significant solvent-exposed region, and we felt that it was well characterized. Approaching the solvent-exposed region, you can have the high potential of maintaining both potency and selectivity, utilizing the solvent-exposed region. But for us, even more importantly, was that there was a handle there that we felt we could really explode a diverse set of physiochemical properties around a set of different molecules and then evaluate the intrinsic permeability. And so we built probably a couple of hundred molecules, with some diverse physiochemical properties. And we were really surprised, actually, that there were no trends. The global data set, we didn't see any trends. That we looked at molecular weight, we looked at hydrogen bond donors, total postal surface area, lipophilicity, the usual factors. There were no trends there. And intuitively, this just didn't feel right. So we took some time and started to pull it apart series by series or sub-series by sub-series. And when we did that, we found one series in particular that had a high correlation of low intrinsic permeability with a particular C-log-P and TPSA space. So there's a lesson there that really spend some time and dig into your data. And I think, of course, in the era of AI and machine learning, that will become far more manageable. This was a program we were executing, in 2016, 2017. A couple of other lessons from there. We struggled for the longest time, maybe not too surprising, low intrinsic permeable molecules, that we had low cellular activity in our workhorse model. And we realized it was an immortalized cell with high efflux transporters. And so that was a lesson for us to move quite quickly to a high throughput PBMC assay and also into a whole blood assay, where we saw about a 10-fold increase in potency. So really understanding how your cellular assays function and what that context is. We ultimately selected the most soluble of those and ran PK. And we identified almost the perfect molecule that had a 0.2% bioavailability, but it was low clearance, and we really didn't want to adopt a soft drug approach, and we can talk about that at some other point. So we were in a great position. We were able to validate preclinically that we had a dose response and a concentration responsive inhibition of clonic JAK signaling, and this was an in vivo mouse colon explant model, and this moved into the clinic, and we were able to establish similar correlations there as well. So a couple of lessons about really digging deep into your data and understanding the nature of the science you're executing on.
Yeah, super fascinating. Thanks for sharing that, Gavin. That's amazing. Yeah, and as we talked about in kind of the pre-discussion, I mean, for every project that succeed, there's, many projects that fail. But nevertheless, there is a lot of learnings that we can get from them. So maybe can you share an example of a, drug candidate that may have failed in development, but still that you took a lot of insights away from that you maybe later applied on other projects?
Yeah, would likely all experience clinical failures. And, certainly I remember one for me that hit really hard. And it's a story about aldehyde oxidase. And, this was work we were doing at SGX in 2008, looking for c-Met inhibitors. And at that time, really, the industry wasn't thinking about the impact of aldehyde oxidase. This molecule moved into the clinic and was terminated almost immediately. It saw some significant renal toxicity in patients. And we were really puzzled. We hadn't seen this in our 28-day rat and dog GLP toxicity studies. And so what was driving this renal toxicology? Subsequently, we went to non-human primates, and we found that we could mimic this renal toxicity, and we found it was due to the accumulation of crystals in the renal tubules in the monkeys. And perhaps even more interestingly, when we started to look at those crystals, they were a primary oxidative metabolite. And this oxidative metabolite was happened to be aldehyde oxidase AOX1. And that was the culprit. And what we hadn't appreciated at the time was the rat and dogs have low aldehyde oxidase activity. So there was a significant lesson learning for us. And I think in this day and age, it's become far more part and parcel of the armamentarium of profiling against aldehyde oxidase. Oftentimes people now use rabbits and non-human primates that are higher expressors. You can use human liver cytosol studies and recombinant aldehyde oxidase. And of course, these lessons now get built in, and we all remember the learnings that we had on hERG and phospholipidosis that had their time in the sunlight. And this was the time for aldehyde oxidase. So it was a really tough lesson for us at SGX at the time.
Yeah, I mean, that's really, really important to know about and kind of dig in the details. So, yeah, thanks for sharing that story, Gavin. Yeah, I want to talk a little bit about industry trends with you in the next few minutes. So what classes of undruggable targets do you think we will be addressing in the next 5 to 10 years?
I would say transcription factors. I think they're going to be really front and center here. Typically they've been a challenging set of targets. They often have low enzymatic activity, very disordered surfaces, few pockets. Sometimes the pockets that are there are cryptic and they don't even reveal themselves. You think of some of the holy grails in oncology, P53s, c-Myc, they've not yet been directly drugged, these two transcription factors. So I think there's going to be an emergence there. And a lot of that is, I think, is driven by some new approaches that are coming to the forefront. People have really started attacking the scaffolding proteins. And so some examples, we saw the recent approval of a menin inhibitor, and obviously downstream of menin, a couple of transcription factors, NF-kappa B and SMAD3. So this is a great way to start indirectly approaching transcription factors. People have also started thinking about cofactors for transcription factors. Can we target those? And so in the Wnt signaling pathway, there are a number of transcription factors, and the recent chatter about approaching this through beta-catenin has really taken front and center there. I think the most impactful area is going to be the utilization of protaxone glues to really approach transcription factors. And we've seen recent progress on drugging STAT6, a highly touted immunology targets, and STAT3 as well in the oncology space, using using Protax. Molecular glues as well. We've seen BCL6 be approached by Boehringer Ingelheim, and a very well characterized IKZF1 approach from a number of organizations, C4 Therapeutics and Nurix. So I really think it's going to be transcription factors are going to be the wave for the next 5 years or so because of the new technologies emerging.
Yeah, thanks for sharing that. Yeah, definitely a lot of exciting areas that you named there. If you could name one therapeutic area that you believe is underserved at the moment, can you name one and explain maybe why this is maybe underserved at the moment?
Yeah, for me, it has to be antibacterials. It's a space I've worked in, but it's always been a bit of a conundrum. There's a continual decline of new antibiotics. We all know there's an urgent global health threat here due to the emergence of antimicrobial resistance. So it's really disappointing to see that. And we all know it leads to longer hospital stays. Myself, I mean, I poster child for this. I went in for back surgery. It was meant to be an outpatient surgery, and I ended up with an MRSA infection, and I was in hospital for 5 days on IV antibiotics. So that extended hospital stay is a real thing. And I think the numbers for mortality each year is around 1 million people die because of antimicrobial resistance. If you ask, we know how to build novel antibiotics. There's been, generations of us being able to do that. But if you look at it deeply, it comes down to the lack of a viable business model. You think about how new antibiotics get used. They're held in reserve, if you like. They only bring the big guns in when everything else has failed. And so if they're held in reserve, that value proposition, the revenue and return on that investment becomes a lot smaller than initially anticipated. And I won't go into detail, but a poster child here was, Achaogen, a highly touted San Francisco antibacterial company, brought a novel aminoglycoside to the market in probably 2017, 2018, and they were bankrupt in 12 months because they just couldn't sustain their operations with the revenue stream they were getting. So it really has to be an area that's underserved and needs some different thinking about how to address it.
I mean, yes, Gavin, I agree with you, and the audience also agrees with you. We have Ryan here also chiming in, and exactly what you said, the incentives are somewhat missing. Yeah, so in the interest of time, I want to ask maybe one more question about your experience with the leadership and team building, and then we'll come to audience questions and to questions that were pre-submitted already. So maybe as kind of the last question from my end, what kind of lessons have you learned, Gavin? You probably managed many large and multidisciplinary teams during your career in drug discovery. Can you share maybe a few lessons that you've learned in those leadership positions?
Yeah, I've been fortunate enough, as you say, to really be involved in leading some of these larger multidisciplinary teams. And I always start with recognizing you have to set a clear vision and goals for your teams, for your organization, such they really have a sight of where they're trying to get to. And it's equally important within each of the various disciplines and the teams is really ensure that each of those have defined and understandable roles in the multidisciplinary teams. Empowerment is a really big piece here. So, we have to rely on subject matter expertise to really bring value to our discussions. And so empowering these subject matter experts, empowering project leaders, to own their decision making, I think is a big part of managing multidisciplinary teams. You have to find ways to have you have to nurture effective communication, I think, particularly in the age of remote work. People have had to find ways to do that even more effectively. So that communication is a real corner piece of enabling multidisciplinary teams to be as effective as possible. Regular cross-functional meetings are really important to make sure everyone is understanding the challenges that each of the disciplines have and how they can bring that together to solve a particular problem. One thing I find is often underutilized is having a robust data management system. And I don't just mean being able to do queries, but really so everyone has access to all the data all the time. So we're making evidence-based decisions across all our teams. And then the final one, and we were talking about this in the pre-discussion, Dennis, was you have to actively mentor and coach your teams. In my career, I've typically set a fair amount of time aside every week, month, and year to coach and mentor my teams, my direct reports, skip reporting. I do a lot of active mentorship, and I think that's a really important piece, so I can bring some of my experience and expertise and help grow new talent.
I love that. And yeah, thank you for sharing your experience today here with all of us, Gavin. That's really amazing, and we all appreciate that. Yeah, let's get to a few audience questions. So we have a live question here from Frederico. He's wondering, what have been the biggest challenges in moving from hit to lead and then from lead to candidate for you?
Biggest challenges. There's been lots of them throughout my career. I think oftentimes one of the challenges, and I'll think about this in a first-in-class space, is not so much the technical aspect around identifying the molecules, but really having the capabilities of assessing those in vivo models, and particularly in first-in-class. Are there really good in vivo models, and how predictable are they in translation? I think that's been a constant challenge for first-in-class molecules, and I'll just focus in on that. Maybe that's a partial answer, but it's not really focused on the discipline of medicinal chemistry. But I find that can be really problematic if you aren't able to pull apart an understanding of your biology with in vivo models, and efficacy models has been a real challenge for me. Less so in best in class, of course, because oftentimes that trail has been blazed by others.
Yeah. Makes sense. Then Ryan is wondering, he's actually a physician, and he's arguing that oftentimes compounds are being pushed further down the line without the target being well understood. And there's often, unintended and unforeseeable side effects. So, to what extent, he's asking, do you feel we might have right-sized target characterization before pushing a candidate further down the pipeline?
Wow, there's a lot packaged in that question. Thanks for that. Um, you know, we all know drugs have moved into the clinic where the molecular target is not well characterized or maybe even unknown, and fruitful therapeutic intervention has been enabled. I really have a strong preference for understanding your target, understanding how it operates, really using that, particularly in the translatability, and really helps with patient selection, patient stratification. For me, those are really important to ensure the highest likelihood of success in the patient population you've chosen. I think this is going to be a massive area for the application of AI as well, that dose this patient, not that patient. And so my strong personal preference is really to really have a good handle around your target. And there are so many good tools out there now to help you understand what it might be. Even there's AI approaches and even experimental approaches. You can do photo affinity labeling to tag your target to get a better understanding. There are structural proteomics and functional proteomics approaches. There's covalent modifying approaches to tag your targets. There are so many ways to really start to understand where your molecule, the molecular target of your molecule. I think that right fit then into the clinic helps with that patient selection. Hopefully that answers your question.
Yeah, thank you. I also want to get to a few questions that we had pre-submitted. One of them by your old colleague at Janssen, Didier. So Didier is asking, is the medicinal chemistry getting distracted by the technology of AI in early drug discovery?
Yeah, I know exactly where you're coming from, Didier. I think the answer is both, yes and no. We've all seen this, voracious appetite for bringing in the shiny new object, which in this instance happens to be AI and machine learning. And I think there are many people I think are overestimating AI's capabilities. That doesn't mean they won't be there in the future. I think we should all recognize it's not a magic bullet. You feed your question into your XGPT du jour, and you get exactly the answer you want out from a medicinal chemistry standpoint. Certainly do not blindly trust AI, but everyone is utilizing it. As I said earlier, I think it's going to be part and parcel of design, make, test, analyze. And if you get away from the hype and look at how it has been used successfully, Didier, there's many real world examples where it's been successfully applied. And my own particular experience has really been in hit ID and lead optimization. And with the appropriate utilization of AI and machine learning, I've seen significant value of using it, but it has to be used appropriately and carefully. It is a tremendously powerful tool for augmenting medicinal chemist thoughts and processes. There is a lot of hype. It can be distracting, but my guidance is to stay focused and recognize the upsides as well as some of the downsides.
Awesome. Yeah, thank you for your take there, Gavin. We have a question that's live and pre-submitted from Ann Tran. How do you view the expanded utilization of human genetic data to nominate new targets? While that's certainly there is value, some targets may have substantial genetic validation, yet do not pan out in the clinic. Most notable, the NAF 1.7 versus the NAF 1.8 inhibition. NAF 1.7 has much more genetic validation, yet NAF 1.6, that inhibitor from Vertex, of course, crossed the finish line. So just your thoughts on genetic validation.
Yeah, yet again, it's not a panacea of all opportunities.
Right.
But I will take the opportunity, really to applaud the Vertex team. The heroic work they did on the NAV 1.8 program. There was genetic validation there that provided strong evidence linked to NAV 1.8 in pain pathways. And so, even though it wasn't as well characterized as NAV 1.7, certainly the genetic validation was there. So I think that it has to be a hallmark of how we think about things, and with emerging technologies such as CRISPR approaches really helps us pull them apart. It's another tool in the toolbox, and the more confidence you have in your target, I think the more likely you are to have success. But it doesn't mean if you always, every target you have high confidence in will pan out. But the NAV 1.8 story is very, very illustrative, even though the NAV 1.7 has been problematic.
Awesome. Yeah, thank you. Another live question here is, how do you see the split in the future between biologics and small molecules? Right now, medicinal chemistry is not too fashionable, isn't it?
I don't think that's true. If you'd
I
asked me
agree. I agree with you, Gavin.
asked me that 5 years ago, I probably would have agreed. People were moving quite aggressively into protein-based therapeutics, and not just antibodies, but ADCs were really hot. Obviously, there is a small molecule component there. But I've really seen a significant upswing in, I hate to use the word popularity, but the utilization of small molecule drug discovery. And I don't know what the numbers were in 2024 of small molecule versus antibodies. I'm sure Dennis can tell us, but I was really surprised to see, if I remember correctly, small molecules, there were more representations than antibodies for what I'd seen in quite some time. Antibodies will always be there. And in fact, small molecules will always be there. And there's lots of people looking at that interface. There's a lot, in particular, cytokine receptor targets where there's significant validation using antibodies. You think of anti-IL-23s, for example, anti-IL-17s, but then people have brought small molecules to tackle that target. So TYK2 modulation is downstream of IL-23 and IL-12. And so being able to bring small molecules to intercept what have traditionally been highly validated targets with antibodies. So there's a play there as well. Lead with an antibody, follow with a small molecule. And many companies have adopted that approach, of course, AbbVie being one of the biggest proponents in that space. So I do see it being actually on the upsurge and not on the downswing.
100% agree with you. And I looked up the numbers while you were sharing this. 38% of the molecules or drugs approved in 2024, so last year, only 38% are large molecules like antibodies.
Yeah. Yeah.
So right on, small molecules are on the rise.
Yeah.
Question. Oh, go ahead.
No, please, Dennis.
No, I was just going to ask another question.
Please, let's have another question.
Yeah, sounds good. Pre-submitted question from Adrian Gill. What is the next biggest field in drug discovery for us to exploit for innovative therapeutic intervention?
I touched on transcription factors earlier. Yeah. I think that's going to be, in my opinion, probably the biggest area that we're going to go into. And I said plenty about that earlier. So, Adrian, I think that's going to be a continually growing space.
Sounds good. Maybe, Gavin, we're actually at the top of the hour. So maybe we close with one last question here from her.
Sure.
Any comments on R&D productivity? How can we increase R&D productivity? And will AI help? And what else could help to increase productivity of R&D?
Yeah, if anyone who's spent a minute on LinkedIn or the blogosphere, there's been huge amount written of the productivity of R&D, and particularly the putative positive impact of the application of AI in accelerating research and accelerating discovery. I think the proof points aren't there yet. But of course, the cycle time for bringing a drug from initiation through to the clinic to get to that proof point, it's a long duration. I do fully anticipate as we start to get some of these proof points, my expectation is we will see an improvement in productivity. Maybe we will be able to bring molecules through the clinic that are improved molecules and have got there at a higher clip than previously. I think there's a significant potential there leveraging AI and machine learning.
Awesome. Gavin, I want to thank you for your time. I mean, this has been a really very nice fireside chat. I learned so many things from you today about those GI restricted JAK inhibitors and, the transcription factor drugging. You shared so many interesting stories with us. So really thank you today from us here at Drug Hunter and from the audience. We're seeing the reactions coming in. Thank you also to all of our audience members who submitted questions, pre-submitted questions. We always love an engaged audience. And yeah, we will be back here next week on Friday this time. We'll be talking about exactly the 2024 drug approvals of the small molecules next Friday. So we hope everybody dials in. And in the meantime, I'm wishing you a wonderful rest of your week and a wonderful holiday on next Monday. And thanks again, Gavin, for sharing your knowledge with us here today.
My pleasure, Dennis, and thanks once again to everyone.
All right. Take care, everyone. Bye-bye.